Good afternoon, everybody. My name is Pablo Abelchauke. I'm a training and outreach coordinator based at HJ Bionet, which is a Pan-African bioinformatics um, community or project based in Africa. Um, and I work in Cape Town, or rather at the central node that's based in Cape Town. And my title is Training and Outreach Coordinator, meaning that I coordinate um, activities for training and outreach, such as webinars. So we have a webinar task force that organizes monthly webinars every last, or rather third Wednesday of the month. Um, we have a webinar, a different one, different speaker from across the globe. And today we have the pleasure of having Dr. Kumar Veerapen, who works at Hale, or rather works for Hale at Broad Institute. And I'll actually now read a bit of his biography so that we get acquainted with him for today's presentation. Dr. Kumar Veerapen manages Hale support and community engagement at the Broad Institute of MIT and Harvard. Dr. Kumar assists in providing health software support using health documentation, forums, and Zulip chat. His role also includes engaging the global community, such as us who are attending now. Thank you so much for attending, into using HEAL as an efficient and scalable genomic analysis tool. Previously, Dr. Kumar was trained in his undergraduate studies. He studied BSc in molecular biotechnology in Malaysia and did his PhD in human genetics and genomics at the University of Miami. He has an illustrious career. You can Google him. He's on Twitter. You can email him. But for today, the title of the talk is HAIL, a scalable tool for genomic analysis, as the title says. And I'm not going to waste much time. Just to say a few house rules. If you have a question, please raise your hand so as to not disturb our speaker. There's an icon on top where you can raise your hand if you want to speak, or kindly type your suggestions, your questions in the chat box, and we'll engage with the questions during question and answer time. So this interactive, interesting talk will be for about 40 to 45 minutes, and at the end we'll have a session for question and answer and suggestions as well as comments. So without further ado, I would just like say thank you so much to all of you for attending, but also introduce the speaker that we've been highly anticipating, Dr. Kumar. The floor is yours, or rather, the mic is yours. Go ahead and give your presentation.
Thank you, Pablo. Thank you for introducing me. Am I clear? Like, can you hear me?
Yes, I can hear you. And for others, you can type
Perfect.
in the chat box. Thank you.
Great. Thank you. So hi, everyone. Thank you for the introduction, Pablo. Blushing a little at a few points. So my name is Kumar. I work at the Broad Institute and Mass General Hospital, where I am community outreach manager for Hale, and I'm also a postdoc at the same time with Mark Daly. So trained primarily as a geneticist. With some statistical genetics background. So the bioinformatics is still relatively new to me. So if at any point later we do have questions that I'm unable to answer, I truly apologize beforehand. But what I will promise is if you can email me that question later, or even if we don't get to some of your questions, please feel free to email me or just send me a personal message on Twitter, and I will get it solved for you. So for today, I would like to introduce this tool called Hail, which is a free open source tool that was developed at the Broad Institute from approximately like 2015. And to start with, just to give you what the outline of my presentation today. Firstly, I'm going to tell you about who we are, and then I do want to know who are you. Um, then I'd like to introduce you what is Hail, why would we use Hail, and how can you use Hail for your analysis, and finally, where can you use Hail on your computer or on the cloud. So the Hail team comprises of all these beautiful people. And I'm Kumar, which is right down there. And I'm the support and outreach manager. Whitney is the person that keeps us on track with everything that we're doing. And we're led by Cotton remotely from the UK right now. And all of us work on a few different things. We're a pretty small team, so we're pretty tight knit. And we work pretty efficiently at trying to get features out there. And another thing to also note is that because this software is relatively new, it's open source, it's highly and actively developed. So sometimes you may see releases that appear once or twice in a week, just to update features, just to update some bugs. And also sometimes we notice that people send multiple requests in a week and we try to process all of them. So we're very communicative with our community. So, as Pabalo was mentioning, I will be trying to make this presentation mildly interactive. So for the first part, I'd like you to tell me where you're from. So where are you? In order to answer this, I'd like you to go to the website above, poev.com/hail 2020. Register your name into it. You don't need to put in an email. And then from there should be an interactive map that you'd be able to ping where you're at. And then this will also enable me to test my geography to see if I get any of these countries correctly. And do let me know if you have any issues with the poll everywhere.
Great. I'm going to also be part of the Africa poll. Dr. Kumar, they're asking that you kindly speak louder. Apparently, some people can't hear you clearly. So if you can raise your voice a little. Sure. Thank you so much.
Of course. Can you hear me better now?
Yes, I can. Let me ask Fatma. Fatma, you are the one couldn't hear. Is it clearer now?
Great. Okay. All right. So I think that's Kenya. Great. Okay. So we have two. I think we have about 14 people here. All right. We have more South Africa. And is that Ghana? I think that's Ghana. Okay. We have more Kenyans. One of my really good friends from undergrad was from Mombasa. And the way she described it was a beautiful city. And I think that's Cape Town. Great. Wow, we have multiple South Africans here. BTW, one of my favorite things from South Africa is your MCC and your Boboti. If you're smiling right now, great. That is Yemen? No, that is not Yemen. Didn't we just have someone tag Yemen? Okay, so we have more South Africans, and I think we may have a Zimbabwean. Cool. All right. We have someone from, let me see, is that Tanzania, if not Mozambique? Okay. All right. Well, thank you for letting me know where all of you are at. It's really important for me to also note this just because I just like to know the distribution of wherever I give a talk to. Wherever I give a talk to, I just like to know distribution of people. To also know that if your schools or that region is interested in using this tool, that I can further make more outreach efforts towards your schools and maybe even come to your schools or your universities to give hands-on workshops, which we do many a time. So moving on, the next interactive slide. Oh, wow, this is not what I expected. Apologies, everyone. I'm having a technical difficulty. Please giggle if you want right now. Great. Apparently my computer doesn't like this right now. Okay, I may need to restart my... So much for fancy technology, eh? All right, there we go. All right, so the next part that I'd like to know. So there's only 3 interactive slides for today. This is the second one. In order for me to also know what tools or align ourselves a little better to our conversation today is what kind of genetic or genomic analysis tools that you currently use. There are 2 ways to answer this poll, which is either using the same website that you were on, or if you're in South Africa, you can use that text message system. My apologies to our Zimbabwean, our other parts of Africa friends, just because the text message number was not available for the other countries. So if you'd like to answer, I'd love to know what are your thoughts on this. And the poll should be active. If you've used Plink, if you use if you use VCF tools. And if you have issues with this, please let me know in the chat. I kept the chat open for now. All right, so I see some people have answered in the chat instead. OpenCGA, Xmizer, GCTA, Plink, and some people have used Hail. Oh, and I noticed that someone mentioned that the poll is still showing the previous poll. Can we attempt to fix this right now?
I don't know if other people are seeing what I'm seeing. I'm seeing blue on your slides. I'm trying to figure.
Yeah, so that's the chat itself. So I was trying to see if there was something people were trying to tell me. Open the chat. Yeah.
Okay.
Yeah. So I'm going to close my PowerPoint for the moment, just because I saw in the chat that some people were mentioning issues with the poll. So I'm going to reopen my PowerPoint. Thank you for being so interactive, team. I'm very thankful that you're that you're giving me that you're telling me if something is working or not. All right, I'm going to try sharing this again, and let's hope this works. If not, we're just going to move on. Okay. Okay, so all right, so this screen is, so people on the chat, can you see the new poll being refreshed on the website? There we go. All right, so I see people have used Plink VP, VCF tools. The cool thing about this app is it gives a real-time update, and I get to also see Plink. I love Plink, by the way. FYI, Hail was developed as a follow-up to Plink. It was meant at that point to replace Plink, but it morphed into its own thing. Max 2, I have never heard of that tool. I'd love to know what that is. SAM tools, BWA, VCF tools. Okay, which does something similar to VCF tools. That's great. VP, I'm assuming for annotation. Snip effect, that is a great tool to annotate your tools, to annotate your variants. Okay. All right. So based on what I can see, the majority of us right now in the audience have used VCF tools and Plink in order to analyze their genomic data. I see SAM tools is catching up, and with SAM tools catching up, this is great. Okay, so let me move on. So based on this, I'm hoping that this next slide, all right, it says activate already on it. You must have possibly read
Okay,
before
I'll try. I'm
coming
just
to this
responding to one of the. Oh, yes, one
of what
already
Hail is.
responding. So I think it's
So
working
based on what
perfectly.
you've read or based on what you know, because you're joining today's talk, what comes to mind when you think of Hail? So just a single word, and if there's issues with it, Pabalo, could you let me know if there's issues with the poll if I need to restart PowerPoint again. Okay. Okay, great. All right, so Hale, when it comes to mind, what is the one word? You can respond up to three words, by the way. Um, Uncertainty. That's new. Large. Version use, need, Python. All right, someone did their reading. Control. Okay, any other words that come to mind? Complicated. I see more words coming on. Okay, so we have Spark as well. Spark is needed in our current version in order to run Hail, one of the dependencies. Calculation. Allele, frequencies. All right. This is likely the most unique compilation of words that I've gotten for Hail Cloud. Expensive. Ah, that is also a concern whenever you do any kind of cloud computing or cloud processing. I can let you know cloud processing is cheaper compared to running it on anything else. All right. Well, thank you for your responses, everyone. Okay, so from what I can tell, it's somewhat an even distribution, and of course, Python is winning. Well, Hail is a Python library, and like it's for the most part, the rest of the words are evenly distributed, and we see that everyone has a certain opinion of this, and that's great. So based on what you're understanding, let's try to, by the end of today's talk, to make Hail a little less complicated, a little more about genetics, and also recapitulating that it is a Python library. And yes, we do use Hail for GWAS analysis. Okay, so moving on. So Hail, in terms of all the abstracts that have been used or published in Hail, this is the cloud that was formed from it. As we can see, some of the words are very genetics related, and some are really not. This is again a shortened version, which is from the abstracts themselves, taken of words which appear more than 4 times. And to start our talk today, this plot was actually made from one of our colleagues at the Broad Genomics core at the Broad Institute, where based on Zachary Stevens' paper in 2016, the question was, is big data astronomical or genomical? As we can see here, a projection from 2009 to 2015, in terms of genomic data, on the top left would be in gigabases of data generated off accumulation of exomes on the first right, genomes and transcriptomes. We would be projecting up to petabytes worth of data. In order to churn through the data with most of the tools that we have right now, it would take a lot more effort, and it's very computationally intense. And to be honest, I don't think most of us would like to wait for weeks at a time in order to run an analysis. So for what we would know most of the time when we run a genome, an exome, or a transcriptome, a genomics data. This would be the GATK pipeline that is generally used, where you would start off on the most left tubes of DNA, RNA, and then the sequencer will churn out reads, which are FASTQ files, and through GATK best practices, following the plot on the bottom for calling variants with GATK, you would have the 3 phases that would then churn out variants. Hail is not replacing any parts of this. Hail comes in right at the end of the GATK best practices here, or the GATK pipeline, by churning through and processing your VCF files that come out from this GATK pipeline. So, what is hail? If you'd like to learn more about hail, please visit our website on hail.is. Our current website is going to be deprecated and will be replaced by a new website in the upcoming few days in our next release, which is a much prettier website. But returning back to this presentation, what is hail? So hail works more like dplyr in R and pandas in Python. Even though Hail historically was developed as a replacement for Plink to handle sequencing files, we have morphed, or rather the users have made Hail less of a Plink and more of a data processing tool. One of the things that we are most proud of is that Hail is an open source library, meaning that if you'd like to edit the code itself on your local computer, you're able to do so. You can also contribute to our features or to changes in our source code. On top of that, the other thing that we have noticed used predominantly is that Hail is not just for large data sets. Hail can essentially work for genomic analysis at every scale. But one of the main strengths of Hail is exploring biobank scale data, where interrogation of biobank scale genomic data has been used throughout multiple biobanks in the world, and it can be scalable to be run on a laptop to 5,000 cores on the cloud, for example, to churn through 1 million genomes. Or right now, what Hail is being used is to process data from the UK Biobank 500,000 exomes, TopNet data, FinGen, and Biobank Japan. Hail also works for modern data scaling because modern data science stack is driven by data frames where modern data science tools such as Pandas and dplyr in R lack data frame scalability. We want to bring efficient use to your data frames by introducing something known as the Hail matrix table that I will go through in this presentation today for effortless manipulation in your genomic data. And finally, the Hail matrix table itself, this input format that we think is the strongest suit in Hail, allows for a unified input platform. You essentially can import every file that is tabular into Hail. Why? Because this data frame extraction allows you to transcend between data formats. Therefore, a matrix table is a format that allows for you to use your VCFs, BGENs, TSVs, GTFs, bed files, and so on. And upon creating these unified input formats, you can then use our Hail Unified Analytics platform to run your analysis. So these are the four main things that what Hail is. And Hail has been used for, well, a few times being published, about 50 something odd times. And in order to cite it, we do have a function within the library in order to pull out the citation method. Currently, Hail has been downloaded for more than 140,000 times because it is a Python library, so you can use PyP. The next slide here may look a little biased, especially to my audience right now. And that's one of the main reasons I'm very thankful that Nikki Mulder allowed me to give a talk or a webinar to everyone here. Because based on this plot, this map here is based on everyone who has published with Hail. And this was based on the first author and the last author on their locations. As you can see, it's very Euro and American centric with little to zero on the southern hemisphere. And that's one of the main reasons that I'd like to introduce you to what Hail is and how you could use Hail to empower your science. And in terms of download locations, so as I was mentioning, Hail has been downloaded for more than 140 times. The map that you see here is colored based on how many times the downloads has occurred, adjusted by the total population size. The lighter the color, the more downloads have occurred in that country. We do see for the downloads, there are some downloads in the southern hemisphere of the globe. However, I'd still like for more people to learn that Hail is not expensive and it's not complicated to run. Once you get the expression language together, which essentially is Python code, you'd be able to run your genomic analysis with so much ease. So why would you use Hail? Because you'd probably be thinking, I have Plink, I have VCF tools. Why do I need to use Hail now? Well, a lot of the times when it comes to running our data, when we look at these different colors here, where the yellow represents the implementation of the tool and the runtime is how long something runs, you don't really have a lot of time for you to think about the science. Sometimes you spend such a long time trying to debug a tool that after you debugged one step, you need to debug the next step. And by the time you're done and you have a pipeline in place, that runtime may be too long because the data abstraction that you have is just not suited to have a shorter runtime. And therefore, you don't really have as much time, or you tend to not have as much time doing your own scientific reasoning. So what we propose, or what Hail has been shown to work for multiple data scientists, especially such as like Konrad Karczewski in the NOMAD data, where they ran everything in NOMAD, 120,000 genomes, whole genomes in a week, and they did all of the processing in less than a month. Even EXAC version 2, where there was about 90,000 whole exomes, was run in the back seat of a car over the span of like 3 days, so that it could be appropriate enough or ready enough for a presentation at ASHG. So what we propose is that once you've learned how to use the libraries within Hail, and we are improving our documentation right now, which is one of my main jobs as the support manager, because our documentation was written previously by software engineers, and now as a geneticist, I know the struggles of reading through documentation. And so what I want, or what we know has already happened, is that the implementation time tends to be shorter. This is variable, but the runtime in general, from what we've seen from all our users, tend to be really, really short. And the reason that's the case is because we want everyone to spend the time doing what you enjoy, questioning what your science is telling you, questioning what your results is implying to you, and inferring from what you have. And a good example, or at least a personal example of mine, was when I joined the unit a few years ago. I was previously using Plink, and I was previously using VCF tools. Some of these tools take a really long time, and things like ANNOVAR that allows you to annotate your exomes and genomes also take a really long time and does not match really very well. My personal experience was I had 4,000 whole genomes. And with the 4,000 whole genomes, a QC that would usually take about a week took me about 30 minutes after figuring out what was my optimal QC parameter. Subsequently, in order to run my annotation using VEP, the variant effect predictor, I ran it in probably an hour. Granted, I use multiple nodes at the same time, and that's another strength of Hail. It allows you to scale your data, and if you have the means to order or to implement your pipeline with multiple computer nodes, that would make things run much faster. And the great thing about running 4,000 whole genomes in such a short span of time is that I could concentrate on what I needed to do downstream at that point, which was to run my linkage scans on whatever I needed to. I had a different analysis plan. So the point of that is, one, I was not biased towards Hail at that point. I was a user, I was a geneticist, and I needed a tool that ran things fast. The tool gave me something that ran things fast. And now, as a community support manager, I see that our users still want things to run fast. Why? So that they have more time for scientific reasoning. So moving on. So Hail, as I mentioned, is a data science library. It's a Python library. And so there's two major facets to it, which is one, how to munge and fling through data. And the first thing that we always have a headache of thinking is, what input format are we using for us to answer a question or to use our tools? Essentially, every tabular format is accepted in HALE. These are some of the genotype-based formats that I superficially mentioned in one of the previous slides. It was not written, but I superficially verbally mentioned it. You could also essentially put in your transcriptome count table into HALE and convert it into a matrix table. And this matrix table can be saved in a smaller size. It's more portable. And on top of that, you can run everything in HALE that you'd likely need to use in multiple different tools. So one such tool when you'd like to sling your data is that when you need to filter through things or group different data points or even aggregate things, such as to compute your mean depth per variance or per samples, whether you want to look at just the heterozygotes, the homozygous refs, or the homozygous wild types. You could also group it based on ancestry labels and sex. Honestly, you could group it in any way possible. If you are accustomed to using dplyr or pandas, some of this may be similar to group by or to mutate. You could also count your TSTV towards your per sample metric. Subsequently, this is one of my favorite things in HALE. Why? Because it can be done so fast. VEP, before I started my postdoc, was a headache for me during my PhD. Now that I have HALE and using the annotation database built-in wrappers, running VEP is so fast, so quick, and so short. And finally, visualization is also relatively easy. This is using a Python library that we've just kind of mapped as a function within HALE. If Python is not your strength like me, you can export some of your data as a smaller table into R, and you can use ggplot or baseplot in order to run your visualization. Because let's face it, ggplot still has the best plots out there. And so when we think of the second facet that HALE gives you and empowers you is the analytical toolbox. So statistical methods for geneticists in order to run their analysis quick and to also get results on the get-go so that you can interpret and spend time on your reasoning. One such really good example that I'd like to point out is the UK Biobank Rapid GWAS that was essentially on every single phenotype that was captured in UK Biobank. We used the linear regression from Ben Neal's group. Specifically, Raymond Walters wrote the script. And if you'd like the link towards how to do this, please send me an email and I'm happy to connect you to Raymond or even the blog link for it, where he computed every single phenotype for only $1 each. And each of this phenotype computed with, obviously adjusted for covariates and resulted in a Manhattan plot and a table that gave him the summary statistics, all being run in less than a day. Subsequently, you could also use things like PCA in order to group your data together to learn what your data is trying to tell you, and in this case, in terms of ancestry for nomads. And finally, we are trying our best to get on linear algebra out there. So from what we know, most of the time LDs are one of the major things that we use to empower our data. And linear algebra methods allow to take this to multiple different levels in the future that we hope to release by the end of this year. So bringing about the input format that I kept talking about, the matrix table. So before we go towards what a matrix table is, perhaps I can recapitulate what a VCF is. This is a commonly used input format, and it looks something like this, where in a tabular format, the columns are all the sample IDs and the metric. The variants are all in rows. The intersect of this would be the genotype and the genotype metric. However, in order to look at a VCF, you need to load in the entire frame itself and search the intersect between sample and variant, which will take a long time. However, with a matrix table, this is so much faster. Why? Because other than just thinking of rows and columns, unlike in VCF, we also have an entries field here. And the entries field is an elegant way to tell you what a cross-section would be. So in this corresponding table right now, I'd like to bring your eyes towards the right of what everything means. So if we were to translate from the input of what we had before of the VCF, a column would be your samples. And these are constant for every row in a data set. A two-dimensional matrix of the entry field, which is the rows and the columns of each of them, is listed right at the end. But moving down from what the right-hand side output was, the rows would be your metric. So all of the sides here would be your metric and also your genotype. So what is your locus, your alleles, your RSID, your quality, your filters, and your info scores. The intersect between your sample and the row fields, such as the locus and the alleles, would result in your metric, your quality metric of your genotype, your AD, your allele depth, your mean depth, your quality, your genotype quality, and your Fred-like score. So all of these are keyed by samples and by your row keys of locus and or alleles. It is so dynamic, the matrix table, that you are able to modify this based on whatever tabular format that you prefer. This can be extended to rare variant aggregation, trio analysis, and transcript expression. So again, we have two different things so in our, sorry, my apologies. So in Hill, we have two different formats, which is just
We can spend 10 minutes. I think that's okay.
What I
Someone's
described
typing.
previously.
But yeah, you
is
can
this
get 10
matrix
minutes.
table that has the rows, columns, and the intersectable fields, which would be the entries. So, Pablo, we have about 10 minutes to spare for me to go through a quick demonstration, or should I wrap up in the next 5 minutes? Okay, so I'm going to exit.
Stop sharing. You can say stop sharing.
Okay, someone is just like, yes,
share
all
again.
right.
Thank you.
So let me, so I'm also happy, so I'm going to demonstrate using a notebook. Now I also have to figure out how to unshare the screen. Ah, okay, there we go. Yep, got it. It was actually hiding behind, something else. Okay, all right. So right now I am displaying Google Chrome, which is my preferred browser. Again, not biased in any way. It just works well for me. And this is my Jupyter Notebook. I'm happy to share the Jupyter Notebook and the set of slides after this to Caballo, and you can run it yourself. So the cool thing about the Jupyter Notebook is you can store everything in your script. It's like a log, except with a lot of really cool annotations. And so why we run Hill in Jupyter is because a few years ago, most computational biologists, which are using command line tools, have started using Python a lot more. And that's why we moved on to this. So the first thing you need to do is to set up your Python environment. So the first step is to import Hail. So you just have to run this line here, which is to import Hail as this object Hail, and also to run some plotting import, so to import the output for showing all of your plots in a few moments. We're now initializing Hail into this environment. And where I've launched a Jupyter Notebook, there are multiple different files. If you have used the Linux terminal before, a Unix-based platform, you'd understand what this line is, which is to list everything within a directory. So this directory that I'm in has a subdirectory called resources. And within Jupyter Notebook, the exclamation point here allows you to use system or Linux platform commands, such as to view what resources I have here. So I have a few different resources within this directory. Why? Because I'd like to use it for some of the demonstration. So this demonstration will be using the 1000 Genomes project, where I've downsampled a small portion of the data so that it's faster and easier to load, of about 20 megabytes as opposed to, I think, a few hundred megabytes per se. So let's explore some of the data using Hail. So in order to explore some of the data, this came in as a VCF file. And we'd like to import the VCF using the function called import VCF. This function is as listed here, where I'm calling on the Hail object with the function import underscore VCF, telling it what file am I importing and how many partitions I'd like to partition the file. So this is a Spark-based concept where you're partitioning your file so that it's easier to load parts of your file. Because honestly, if you load one large file at just one partition, it will take a long time. But if you're loading a little bit and piece of your file, it would be a little faster to access parts of your table. So upon importing, I'd then like to write it out as a matrix table. So as I mentioned, the matrix table is a file format that is native to Hail. So this just imports VCF, exports it out as a matrix table that can be read later on. So you'd have to convert your VCF files into a matrix table. So once this has run, we can then import the matrix table into Hail, and that's really fast. So that import is done, and we can explore what each of these things are by using this function, .describe. I love the argument in .describe, which is called widget equals to true, because you can interact with what is a row, such as locus, alleles, RSID, quality, filters, and info, what is a column, which is your sample, and you can also see what methods are commonly used to give you a clue on how do you run a certain analysis. And obviously, if you have any questions about this after this presentation or after running this yourself, you're welcome to email me, or I'll put you in contact with someone who would have the answer for you. The most appropriate thing is to go on our Discuss. Entry would then be the metric that I was mentioning and the genotype calls. So you can either click on these tabs to view them, or you can click on the widget itself. This is one of my favorite features. So you can also see what's within your file by looking at your samples. So show me the sample IDs, show me the locus IDs, and show me the genotypes. So calling on the matrix table, followed by what you want to show, and do you want to show it or not? You can also use Summarize, which gives you a really cool overview of what your object is. Or in this case, the object is your matrix table. How much is missing, how much is non-missing in your depth metric, or in your AD, which your allelic depth. You can also count, which gives you the number of rows and the number of columns. So the number of rows here would be the variance, and the number of columns here would be the samples. So there are a few functions in Hail that allow you to run your genetic analysis, because some of this may seem a little like generic. We do have more specific terms like, let's summarize the total number of variants in your matrix table. So you're using this function to call on a summary table like this. So the summary table, as we saw before here, we have 13,000 variants. In your 13,000 variants, how many alleles there are per variant, what is the counts by allele type of Smith's indels, your transition transversions, and the ratio is also churned out for you and grouped immediately. Your variants per contic, where contic here would be chromosome. And so, as I was mentioning at one point today, the annotation using Hail is super easy and super fast. So we're using an annotation table here where, as I mentioned, we're using the exclamation point would escape from the Jupyter Notebook and would use the system command. And this looks at the annotation file and what it looks like. So this is a table of columns, the sample ID, to map onto my matrix table, the population type, the superpopulation of the ancestry themselves, whether the person is female or male, the person has purple hair or not, because I thought that would be fun. How many people have purple hair? Caffeine consumption, and who have polydactylism of the feet, 6 toes. So we can import this instead as a table. I had one picture at the end of the slides before describing the difference between the matrix table and a table. The easiest thing for me as a geneticist when I first started learning the difference between these two is I would use tables for annotations and I would use matrix tables for genetic data. So in this case, I'm importing the annotation as a table. You're imputing every column. So the impute here means what data type is sample, what data type is population. So Hale would infer or impute whether sample is a string or an integer, and you'd key everything by the sample ID. So these are the rows. So we can run this. It's pretty quick because it's pretty small, and you'd like to see it in your tabular format. So as you can note here, the top line is the same exact header that you mentioned before. This gives you the data type of strings, booleans, floats, or boolean. And if it's wrong, you can edit it in this area here. You can also summarize this. Summarize, the only caveat of summarize is if your data is pretty large, it will take a lot more time for it to run. So this is relatively small. So I suggest using something like show and picking certain things to show as opposed to using summarize as a whole because large data sets will take much longer. So this gives you a summary for each of those columns that you see keyed by your row that is your sample. So let's annotate our matrix table. In order to annotate, we're annotating on each column. So each column would have a new annotation here, and that's it. We've already annotated it. And we can use this widget, the describe, in order to see where it is. It is now saved as Pheno. Every header that we saw just now is all here within your columns. So I like this because you get to see where everything is. Frankly, I think most geneticists are very visual people. So let's query the phenotype fields. So this query tells me what fraction of my data have purple hair. And this query is give me the distribution of people in these superpopulations of Africans and so on and so forth. The sample QC is one of our coolest functions because you're able to QC your samples, and it would appear I apologize, yeah, it will appear right here in your columns. So it gives you the depth stats, the genotype quality stats, essentially taking every metric that's possible in terms of your QC metric and process it for you, saves it as a column. And let's view the sample QC, what we just established, what we just saved into our matrix table, and look at the depth mean and the call rate. So in order to do this, we can plot, and again, plotting is easy. It's not as pretty as R, but this is using Python, and you can see what the call rate is like. So we could essentially cut off your call rate at approximately here, or even higher, and not lose a lot of data. You can also filter your columns using the generated QC statistics. There are 343 columns that we established before. And if you'd like to QC by removing everything higher than 95, removing everything lower than 95 and only keeping everything higher than 95, we have now removed about 20 samples because those 20 samples just didn't fit in our first metric. We can continue QCing. That was just a sample QC, and this is the variant QC. So in the variant QC, we'd be able to see it in our rows. So we'd be able to see the DP stats, so on, so forth. We can show our QC stats, and we can plot our QC stats. In the interest of time, I'd just like to jump. Okay, in the interest of time, just like to jump to here. And we can plot again. So this is just plotting your variant QC of a call rate of more than 95%. And now we can count and we can write it
Thank you
out
so
at the
much.
matrix table all
I
over
think you
again.
still have a presentation going,
So
right?
that is my demonstration.
Because there was a
in
question
nutshell.
from
I did
Dean
have a couple of lines that I
asking
had
about,
in addition to
let me
this.
check.
But
Did you want to
I
raise your
have
hand
no
and
idea
ask the
why
question
it's not
yourself?
appearing right now.
Is the
So
exporting to
I'm just going to leave it as that.
matrix table
because we're
step
almost out of
generally
time.
a big bottleneck in a workflow?
And
that is a question from...
I'd like to wrap things up.
Yes.
Yeah. No, actually, so who is that from? That's from D. Todd. Um, so is exporting a matrix table generally a big bottleneck in a workflow? No, it actually isn't. It's one of the fastest steps because importing takes really quick. The only part that would take a long time is deciding on the number of petitions that you want. So I have, this is something that I learned at the start when we had like our first version of Hail. One genome would be one partition just because like a genome would, one partition per genome processes much faster. We don't really have like a exact math to it. It's really rather random and abstract. Can you please also share a link to the sample data to accompany the notebook? Thank you for taking note of that. I will also, so what I will do, Ibra, I will give you the Jupyter Notebook, and I will also update the Jupyter Notebook with some lines of code that accompanies that I wanted to show as well today. That is our linear regression. So how would you run a linear regression? How would you run a simple QC? And how would you finally plot a Manhattan plot? I find that these are all some of very, very important questions that we ask at the end. And I will share that in addition. I hope that's okay, Ibra. Those will be probably 3 additional line of codes on top of what I've demonstrated today. You're very welcome. And if I forget, I'm sure Pabalo will remind me. He's extremely efficient that way. So if you have any other questions, please feel free to ask. Can you please also share how to run an Ancestry PCA? Sure. What I can do is, Melissa Nell, what I can do is I will include a line on generating PCs, if that's okay. Or the question was, did you want a PCA plot? And to Bright, yes, of course.
Raise your hands if you want
I
to
think
speak.
Pabalo will be sending out
There's an icon
slide
on top there where you
slide
can
deck
press
and the Jupyter
and we
Notebook
can
to
hear you speak.
everyone.
Alternatively, continue typing in the chat box.
Generating PCs would be great.
Aiton wants to
Okay,
speak.
so the
Go
3
ahead,
additional
Aiton.
lines of code that I was mentioning to Ibra earlier included generating PCs. Okay, so Aiton, is the matrix table format readable by other software? No, it's a Hail native format. I know you're probably wondering, oh God, another format. But I'm telling you, with matrix table, it really makes your life so much easier because all of the formats and most of the tabular formats out there is compatible to be exported as a matrix table. You're welcome, Aiton. while everyone is typing, I just like to just splash through a few screens for you. So we can run Hail on a laptop, on a server, on a high performance computing cluster, or preferably on the cloud, where the installations are all here. We don't support HPCs such as Slurm, Uger, and LSF, just because it doesn't work as efficiently. But the entire instructions on how to do it is on our hail.is website. And the first thing you need to know is just pip install Hail. But yeah, any other questions? I love that. Is it difficult to set up Spark? No, it's actually, it used to be much harder. Let me put it that way. It is so easy right now. If you have issues with your installations, by all means, we at
Yeah,
the
so
team
I
are
actually have a
very
good
frequently.
experience running
We're moving
Hail
away
since it
from
was
Zulip Chat,
launched
which is like an
version
additional IM
1.2.
that you can install on your
So that's about
cluster,
2 years ago. So I
into
used it many
Spark.
times
And the reason is so that people
especially
can search through
to
things
get
much
more
easier.
detailed
And no question is too
allele
easy or
frequency
too hard to.
calculation, that's including the number of heterozygotes and I did that
Okay,
for the
Mohammed, you
whole
wanted to speak about your
African
question?
only subset of the 1,000 genome data. And I also used it in more like in my local Sudanese population data as well.
Okay.
I also and I recently used it to do
Yeah.
plotting of GWAS QC, for example. We had a lot of imputed data, millions of 92 million variants in a set, and I used it to plot uh,
Okay.
the imputation score histogram. So it is very valuable. I like it very much. And my question is actually, would you be adding like support for mixed linear models
Hmm.
and genetic relatedness matrix in the near future?
Okay. Ah.
Okay, sure, we'll do that. Yeah.
Okay, thank you, Mohammed. That's one. Thank you for the compliment that Hail has worked really well for you. Uh, one thing is, if you have published that, can you please send me a link so that I can include a part of Africa in my map of everyone who has published in Hail? And secondly, yes, please. Oh, by the way, if anyone wants to edit that page, if you publish using Hail and I just not like included it, by all means, you can edit that page yourself as well, by the way, because everything is open source. Okay, so to answer your
Yeah,
question,
yeah, yes,
Mohammed,
yes, it is
mixed linear
our package.
models,
Yeah,
we currently do not support mixed models in Hail. And the reason is because our linear algebra has been having a lot of issues trying to implement it. What we've done instead is we've actually suggested to people to run using Sage. I don't know if you've heard of Sage, Saige. It is a really cool, yeah, yeah, that's an R package. We may be releasing something that allows you to what's the word, to communicate with
so I'm
Sage,
actually,
not at the current
yes, I'm
moment.
actually
So I
subscribed to the Hill
apologize for
discussion.
that. We
So I
don't
received
have full
weekly emails,
support for mixed
which is very
models
nice.
as yet.
But
It is
I would,
on
yeah,
our
in
features list.
while we have you, it would
But
be
yeah,
very
just
nice
keep
if
an
you
eye
would
out
add
for it.
more
And if at any
documentation
point
on
you feel like we're too quiet on
how
it, just send us a bullet
to
message
interpolate
or
with
just post
SparkML
on
library.
discuss.hill.is to see what our status is
That would be,
in
yeah.
like 3 months if you don't hear from us. Good. Yeah.
Yes.
Oh, that's a good note because I don't think I've actually gotten that comment before. Uh, let me. Sorry, I have to grab like a post-it right now. Spark ML, right?
Thank you. Thank you very much.
Got it.
Yes. One more
Okay.
question. Aiton, you
Thank you
can
for the
go ahead,
comment.
please.
I love
Go ahead
taking
and ask.
comments
And
at any point. If you have anything
Hi,
else, like
hi,
I'd love
Kumar.
to
Thank you
hear
very
from
much.
you. So, like my
Just
email address is
a
also
sort
over
of
here.
very broad
Feel free to send me an
question.
email at any
If we're going
point,
to
and
have
I'll
a lot of
get
genotype
back to you in
data
that
and a
regard.
lot of samples, well,
Yeah.
probably
So
not
Mohammed, thank you
broad
for your comment. Thank you for your
numbers, but
question.
thousands of samples and many genotypes.
Okay. So I think Aiton
What is
has a
your
question.
suggestion on, and we're going to want to use Kale to calculate things periodically. Am I right in thinking our workflow could be to make a big MT file, and then people can work off that as they need to? And we build that new MT file as we get new data. Does that make sense? Both. So we're trying to combine as many of the African data that we have together, and then we'll periodically have questions that different people want to ask.
Yeah. Okay. So if I understand that question, you have thousands of whole genomes or genotype data. Okay. So you have yeah.
Yeah, I'm just asking, I mean, sort of more in a pipeline sort of point of view. So we won't be continuously adding, but periodically we would be. So
Okay.
maybe once a
But are
month
you
we'll be
going
updating
to
our
continuously
collection of
add,
everything we have.
continuously add more genotype data onto your
Is that the sort of flow you
onto your
guys work
matrix
with?
table, or is it just that you want to send one matrix table out to multiple people to run analysis at different times? Ah, okay. That's a great question, Aiden. Yeah. So, thank you for asking that question, Aiden. We do. It's actually one of our major requests last year, our feature request last year, to come up with a VCF combiner. So to recapitulate Aiton's question, if I understand correctly, is that periodically, so Aiton has X number of whole genomes, and periodically he'd like to add more whole genomes onto this data set. So instead of doing a whole joint caller all over again, doing the calling all over again and combining it from like the VCF point, can he just do it from the matrix table itself? Yes. So as long as these files are exported first from your GATK as a GVCF, you
It
can
wasn't
use
a very
something
specific
known as
question.
the
It was,
VCF
so
combiner.
yes, you
The
definitely
VCF
gave me some
combiner
pointers. Thank
is
you.
our Hail joint caller. And what it does is every single time you have a new GVCF come in, you can combine it onto your matrix table. So as one, to save time, and two, to save money from running the pipeline all over again. Aiton, did that answer your question? I hope that was what you were asking. Got it. So yeah, VCF combiner is what I would suggest for you to use. VCF underscore combiner is listed right now as an experimental function.
Thank you so much
It
for
still has like
that.
a few bugs here and
I'm
there,
looking
but for
at
the most
time,
part, it is
and
pretty
I
easy
just want to say thank you so much. So firstly, I've
to run.
recorded
And
the
again,
presentation,
on
so I'll
discuss,
send out the
just
recording
send us a
through our
question if
YouTube
you
channel,
run
because we have a
into
YouTube channel. I'll
any
give the link,
problem at
the
all,
resources and
and we will
slides
answer you. Our
to everybody
turnaround
who
time is
attended
actually
this
less than
webinar. So
12
thank you so
hours.
much to
Sometimes
the
I wonder if
participants.
any of
But
the students.
mainly, Dr. Kumar, I want to say that this is one of the most
Okay, so
interactive webinars that I've ever had at the Bayonet. The demonstrations,
yeah.
the polls, it was very interesting and interactive, and we really appreciate it. If you have more questions for Dr. Kumar, please follow him on Twitter, please email him, and he'll gladly be able to assist. So yeah, I just want to say thank you so much, Dr. Kumar. I don't know if you have anything else to say.
آخر لحظة.
Thank you so much. Okay, cool. Everybody enjoy your day,
Just go
and
in
see
and
you
scroll
next
through on your
month for
computer,
the June
and
webinar,
if at any point
which we're
your
busy organizing
university
at the
would
moment.
like
But
us to
Dr.
come
Kumar,
down
this was
after the
really,
pandemic
really
to stay
interesting,
safe,
if I say
we
so
are
myself. We
happy
really
to
appreciate
fly
it.
over.
Enjoy
We'll
your
figure
day.
out the logistics and cost of it later on offline, but like if